Hi everyone, this is Dr. Ujwak Neuro Academy. Today we'll be talking about the functional units of the cerebellar cortex. The cerebellum is made up of outer gray matter, or what we call the cerebellar cortex, and an inner white matter. The white matter is formed by afferent and efferent nerve fibers. The gray matter is made up of cell bodies or dendrites. There are gray masses called deep cerebellar nuclei that are located within the white matter. Now, the gray matter or cerebellar cortex is made up of structures that are arranged in three layers. The three layers of gray matters are the outer molecular or the plexiform layer, the Purkinje cell layer, and the inner granular layer. Beneath these cortical layers, in the center of the cerebellar mass, are the deep cerebellar nuclei that send out signals to other parts of the nervous system. Now, the molecular layer is the outermost layer of the cortex that contains stellate cells and basket cells. The Purkinje cell layer is situated between the outer molecular layer and the inner granular layer. It contains the flag-shaped Purkinje cells. The dendrites of the cells are sent through the entire thickness of the molecular layer and arborized there. The granular layer is the innermost layer of the cerebellar layer that is in between the Purkinje layer and the deep cerebellar white matter. It is formed by intact neurons called granule and Golgi cells. The cerebellar cortex receives afferent signals from other parts of the brain through two types of nerve fibers, which are the climbing fibers and the mossy fibers. The climbing fibers arise from the neurons of the inferior olivary nucleus that is situated in the medulla and reach the cerebellum via the olivocerebellar tract. The inferior olivary nucleus relays the output signals from the motor areas of the cerebellar cortex and the proprioceptive signals from different parts of the body to the cerebellar cortex via the climbing fibers. The proprioceptive impulses from different parts of the body usually reach the inferior olivary nucleus through the spinal cord and via the vestibular system. After reaching the cerebellum, the climbing fibers are sent into the molecular layer and terminate on the dendrites of Purkinje cells. While passing through the cerebellum, the climbing fibers send collaterals to the deep cerebellar nuclei so that the impulses from the cerebellar cortex and proprioceptors of the body are conveyed not only to the cerebellar cortex but also to the cerebellar nuclei through the climbing fibers. Each climbing fiber usually innervates one single Purkinje cell. Unlike the climbing fibers, the mossy fibers have many sources of origin, namely from the motor areas of the cerebral cortex, the pons, the medulla, and the spinal cord. Fibers arising from all these areas send collateral to the deep cerebellar nuclei before reaching the cerebellar cortex. Some of the mossy fibers also arise from the deep cerebellar nuclei. Now, mossy fibers reach the granular layer of the cerebellar cortex and divide into many terminals. Each terminal enters a special structure called the glomerulus and ends in a large expanded structure that forms the central portion of the glomerulus. The dendrites of granule cells and the axons and dendrites of Golgi cells synapse on the mossy fiber, giving a thick bushy appearance. The functions of cerebellum are executed mainly by the impulses that are discharged from the cerebellar nuclei. However, the cerebellar cortex controls the discharge from these nuclei via the fibers of the Purkinje cells. It is usually achieved by the signals received by the cerebellar cortex from different parts of the brain and the body via the climbing and mossy fibers. Now, the climbing fibers excite the Purkinje cells directly and the cerebellar nuclei via the collaterals by releasing aspartate. The excitatory effects of climbing fiber on Purkinje cells is very strong because each climbing fiber ends on a single Purkinje cell. The mossy fibers excite the Purkinje cells indirectly. In the glomeruli, the mossy fibers release glutamate and excite the granule cells and Golgi cells. The collaterals of muscle fibers activate the cerebellar nuclei also by glutamate. The granule cells, which are activated by muscle fibers, in turn excite the Purkinje cells. It also excites the stellate cells and the basket cells through the parallel fibers. The neurotransmitter utilized by granule cells is the glutamate or aspartate. The granule cells are the only excitatory cells in the cerebellar cortex, while others are actually inhibitory cells. Each muscle fibers innervate many Purkinje cells indirectly via the granule cells. So the excitatory effects of muscle fibers on Purkinje cells are actually weak. The stellate cells and the basket cells, which are activated by the granule cells, usually inhibit the Purkinje cells by releasing gamma aminobutyric acid. The Golgi cells that is activated by the muscle fibers in turn provides feedback inhibition to granule cells by releasing gamma. That is, it inhibits the transmission of impulse from muscle fibers to granule cells. The cerebellar nuclei are actually excited by collaterals from climbing and muscle fibers. In turn, the nuclei send excitatory impulses to the thalamus and different nuclei in the brainstem. However, the signals discharged from Purkinje cells inhibit the cerebellar nuclei via GABA. The Purkinje cells inhibit the activities of the vestibular nuclei also. Thank you for listening. Please like and hit the subscribe button.